the work I'm going to talk about today is joint work with Yoav Gilad's lab in Chicago, looking at using human iPSC-derived cardiomyocytes to model drug response and side effects. And while this is a specific use case, I think it's a nice demonstration of two things. One is that human iPSC-derived differentiated cell types are a really cool model system to think about using, especially for perturbations. And also that there are settings where it's really hard to collect the scale of GWAS data you'd like to get, and that doing EQTL type analyses can actually get you some real leverage. So I wanted to give a little bit more background about myself, and this is maybe particularly relevant for this seminar, which is, as John said, I did my PhD in statistical machine learning with a pretty Bayesian tint. So one of the main areas I worked in was this realm of methods that fall under Bayesian nonparametrics. So if any of you have interacted much with Tamara Broderick, you might be aware of some of these. The idea with Bayesian nonparametrics is to try and have statistical models that can adapt their model capacity to the data at hand. So the most canonical example would be something like a cluster analysis, or you're trying to fit a mixture model, and you'd like to have a principled mathematical way of working out how many mixture components or how many clusters you should use for a specific data set. And Bayesian nonparametrics gives you that well-founded, that solid mathematical foundation to do that. So I worked on Bayesian nonparametric methods for sparse factor analysis, so sort of analogous to principal components analysis, as well as for spatial temporal data, network analysis, particularly social network data, and Bayesian hierarchical clustering. I also worked on a class of methods which fall under variational inference, and these are usually deterministic algorithms which offer alternatives to Markov chain Monte Carlo, which is sort of the most common thing you'd think of doing if you're trying to do inference in Bayesian probabilistic models. And the work I did in this area was to extend these methods to be applicable to a wider range of probabilistic models. And then during my PhD, I got increasingly interested in applications of statistical machine learning in genetics and genomics. One of the first projects I got involved with was looking at epigenomic changes during the development of cardiomyopathy with Roger Fu's group. And then a little later, helping out with the Mother Consortium, which is a really nice cohort of large-scale cohort of female twins based out of the UK. And they were one of the first studies to have large-scale observational data with gene expression across multiple tissues. So we were able to look at gene expression changes both with age and with across tissues. Then during my postdoc at Stanford, I've worked on different ways to try and get more information out of functional genomics assays, so mostly focusing on RNA sequencing. So work I did with Stephen Montgomery and Alexis Battle was looking at using allele-specific expression to improve power to detect gene-by-environment interaction effects on the transcriptome. And more recently, with Yang Li, who's now at Chicago, who was in the Pritchard lab, looking at new methods, new ideas on how to quantify splicing, RNA splicing variation from short-read RNA sequencing data using an algorithm we called leaf cutter. So the work I'm going to talk about today is actually going to leverage both of these tools, and you'll see how. So to give the setting the scene a little bit for why it's interesting to look at anthracycline cardiotoxicity. So this seminal 1979 paper showed that as you increase the chemotherapy dose, the lifetime probability of developing chronic heart failure goes to these really high levels. And what this graph doesn't show you, which I think is one of the most sort of frightening things about this, is this can occur years or even decades after the chemotherapy, which is particularly tragic for pediatric cancer patients. So as well as, these heart side effects are obviously terrible in themselves, they also put these limits on the chemotherapy regimen that you can use as well. So as you might imagine, there's interest in understanding why some individuals do develop these cardiac side effects when exposed to anthracyclines and some don't. And because of that, there have been several attempts at genome-wide association studies to look for genetic variants that seem to confer resistance or sensitivity to these side effects. So I think especially talking here, you'll notice that the GWAS, the sample sizes here are somewhat pathetic. So this 2015 Nature Genetics paper looked at a small pediatric cohort, and they found one sort of nominally significant, actually non-synonymous coding variant in this transcription factor. But follow-up studies have been less than conclusive. So this 2016 paper used a larger cohort, found no genome-wide significant associations, did find that they took a small number of their top associations, even though those weren't significant in the discovery cohort, and tried to replicate them in further cohorts, and could find for one variant, we're able to show very modest replication in 2 further cohorts. And the most recent paper, again, modest sample size, found no genome-wide significant associations. They did check for whether they see an association with the finding for this variant from the 2015 paper, and they're like, oh, well, this is close to significant. And then you look at the effect size, and it's the opposite direction to this first GWAS. So the reason this field is really struggling on the GWAS front, right, is that this data is all coming from clinical trials. So you can't go and do this in biobank, because it's not like everyone in UK biobank got exposed to doxorubicin. It'd be a very small number of individuals. So the idea we had was to, instead of looking at ACT itself, to look at transcriptomic response to doxorubicin and see whether that could give us a bit more leverage on the problem. Yeah.
So I'm clear, the GWAS is taking people who have been exposed to this drug and asking of these people which of them you have cases and controls for who had cardiac events during the course of the trial, and you're just trying to.
Yeah, exactly. So that's why the sample size is so small, is because it's only the people who were signed up for the clinical trial or who got the drug. Yeah. So what we did in collaboration with the Gilead lab, and when I say we, all this is really, all the experimental work was done by Courtney Burrows, who's now graduated from Yoav's group, was to take blood draws from 45 individuals from the Hutterite population that's been extensively studied by Carol Ober's lab, extract lymphocytes, make lymphoblastoid cell lines, and then reprogram to induce pluripotent stem cells, and then differentiate to cardiomyocytes. So that gives us a panel of 45 iPSC-derived cardiomyocytes, and we can then take those, expose them for 24 hours to differing concentrations of doxorubicin, and then do RNA sequencing to see what transcriptomic changes have occurred as a result of this exposure. Again, I should point out this is a huge amount of work, right? Like maintaining 45 cell lines and doing all of these exposures and then library prep. So I very much appreciate Courtney having done the hard work on the experimental side of things here. We see really strong and robust gene expression responses to doxorubicin. So this is showing principal components analysis across all of the samples that we have, starting out at baseline, this initial lowest concentration, these are all at micromolar. What you already start to see here, and I should say lines here are connecting samples from the same individual. You already begin to see variation in the response between individuals. So notice that this intermediate 1.25 micromolar samples, depending on which individual you look at, for some individuals, that concentration looks more like the 0.6 micromolar, whereas for some individuals already at the 1.25 micromolar sample, you've already gone the whole way through this trajectory to pretty much this endpoint, suggesting that those individuals are responding more strongly to doxorubicin. If you do any sort of reasonable differential expression analysis, you have a sample size and such a strong gene expression response that basically all genes are differentially expressed, which is not particularly interesting in itself, which means that to do anything downstream, we've thought what would actually be more useful would be to cluster cluster genes according to their gene expression profile across concentrations. So we use a mixture model approach to do this, and using some of those ideas from Bayesian nonparametrics, we end up choosing to use 6 gene expression profiles. So these have distinct patterns across the concentrations. The largest, cluster 1, for example, shows continuous downregulation with increasing doxorubicin concentration. But then you see other patterns like cluster 2 here, where there's an initial increase in expression in response to this lowest doxorubicin concentration that we use, but then downregulation at higher concentrations. Having put all of our genes into separate clusters, we can do functional enrichment analysis. I don't want to focus on these too much, but just to say, and this was done using Homer, I should say, you see some reasonable things, like it turns out actually Homer in its default set of pathways actually has a gene set which was derived from looking at doxorubicin exposure. And these are genes down, the DN here is for downregulated. And so you'll see the clusters where we have initial downregulation of expression in response to doxorubicin are the ones where you see enrichment for these gene sets, so that's encouraging. Another interesting one here is that targets of p53 are enriched in the clusters where you see an upregulation of expression at that initial doxorubicin concentration. That's interesting because one of the main mechanisms of action of cellular damage that doxorubicin produces is oxidative stress, and p53 is responsive to oxidative stress. So I think here, I probably don't need to say too much about what expression QTLs are, but it's going to be important for a lot of this, so I'll just go over it briefly. So usually the mental model we have of what's going on with an expression QTL is that there's some transcription factor, maybe at a promoter, maybe at an enhancer, that's binding some genomic element. Through cis-regulation, that transcription factor changes or regulates expression of some local gene. There might be, the fact that we have an eQTL suggests that there's a regulatory SNP that results in differential binding between individuals of that transcription factor. The example I'm showing here, the G allele is resulting in increased binding of this transcription factor. I'm imagining this is an activator, so it's resulting in increased expression of this gene. Of course, for better or worse, humans are diploid, so what actually is happening is that we see usually sort of the average effect across the 2 alleles. So this individual being heterozygote means that one of these alleles is being expressed more, whereas the other one isn't, and what we get to see is the average of these 2. Response eQTLs, I think, are most easily, so what we're really interested in looking for here are genetic variants that seem to modulate transcriptomic response to doxorubicin. And so I think it's maybe easiest to show what one of these looks like. So this is showing gene expression, log CPM, for this mitochondrial gene, the different doxorubicin concentrations broken down by the genotype of this cis SNP. And this is one of a handful of examples where we see this really strong modulation of the response by this genetic variant. So here you see that for the major T allele, you see an initial downregulation in response to doxorubicin, but at higher concentrations, you actually see higher expression. Whereas for the minor allele, you see a completely different response, right? There's almost no change at the 0.6 micromolar, but as you increase concentrations, you see a strong downregulation of expression. This is, as I said, there's a handful of examples we find like this. For the most part, what you see is more subtle modulating effects rather than changes in sign. So mapping response EQTLs poses a few challenges relative to regular EQTL mapping. The first is that we have repeated measurements from the same individual. We use the Hutterite population, so the 45 individuals we have are actually related through a known pedigree, so we'd like to be able to take that into account. We always, with these big RNA sequencing cohorts, worry about latent technical confounding, but we'd like to keep everything computationally efficient as well. So the approach we end up using is a linear mixed modeling approach based on ideas from a really nice paper model called Panama, developed by Niccolo Fusi, Ollie Stiegel, and Neil Lawrence a few years ago. Their model is called Panama, so our model is called Suez. So it's a linear mixed model, which means it's going to have fixed effects and random effects. So Y here is some normalized gene expression. We actually use quantile normalized gene expression, so we're not too worried about outliers. So N here is going to index over individuals. C is going to index over concentrations, and G is indexing over genes. So the first thing we're going to include in the fixed effects are the effects of genotype. So D here is dosage. How many copies of the minor allele do you have at the SNP, the regulatory SNP that you're testing? Beta CG here is the regression coefficient, so the EQTL effect size. And in I'm showing you here the sort of full model that you could think about, where that effect size is dependent on both the concentration, and of course it'll be different for different genes. We also need a fixed effect for the concentration, because as we saw, there are these huge changes in expression depending on what concentration we're at. Then the random effects. So this is actually maybe the most interesting term in the random effects, which is there to deal with technical confounding. And this is analogous to a factor analysis model. So if you've heard about or used models like PEER that try to remove technical confounding, the idea is very much the same, that you have this matrix factorization. Here things are handled a little bit differently because we're going to think we have factor loadings and latent factors, but we're actually going to end up integrating out the factor loadings, which is actually sort of the opposite of what you'd normally think about doing in factor analysis. And I should say both of these are going to be, both of the W's, while we're going to integrate over the X's, integrate over the W's, we're going to have to learn the X's. We then have a random effect for the individual, because we have, and that's to deal with the fact we have repeated measurements from the same individual. And then we have a term that's going to account for the relatedness between individuals. Finally, we have isotropic noise. We need to specify variances for all of the random effects. So I'm just showing those here. All of these are just the Gaussian distributions you would expect. The only interesting one really here is that we need to include this known kinship matrix between individuals. So the first step of Suez, and this is analogous to what Niccolo did with Suez, is to learn the random effect variances. And this is going to be done across all genes simultaneously while ignoring genetic effects. So we're going to assume that the genetic effects are negligible from the point of view of learning these random effect variances. So that corresponds to setting these beta coefficients to 0. If you then integrate over the random effects, you end up with this multivariate normal distribution where the fixed effects give you the mean term, and I'll explain what V is in a second, and all of the random effects specify your covariance structure. So this V matrix here, so rows of V correspond to different samples. A sample is an individual concentration pair, and VSC is 1 if that row is at concentration C, and otherwise it's 0. The the covariance matrix here. So this first term comes from that matrix factorization term with the W's integrated out. And so we're going to have to learn both the sigmas here, which correspond to the prior variants of the W's, as well as the X's, which are the latent factors themselves. Then this term results from the individual random effect, and that means I need to say what U is. So U here is a matrix where an element is 1 if that row corresponds to individual n. Then this term comes from the dealing with relatedness between the individuals, and finally noise. So we optimize the random effects, the fixed effects, and the random effect variances, as well as these latent factors jointly across all genes. And this is done using LBFGS with the model implemented in the Stan R programming language, which I'll mention a bit more in a minute. So in the second step, what we will do is fix a global random effect covariance using the estimates of both the random effect variances. Yeah.
Did you have a prior on the X's as well?
No, so we don't. Well, we do actually all of these are like very gently regularized, just but more for numerical stability than any sort of statistical concern. Yes, you certainly could think about doing that. Here, I didn't do, there are, going back to the Bayesian non-parametric stuff, there are, and I worked on, as I said, Bayesian non-parametric versions of factor analysis, which are also effectively regularizing both the factor loadings and the factors themselves. Here, we just use some heuristics to set the number of latent factors we used, which is also sort of a regularization in itself. But, yeah. Yes?
Can you give an intuition for what the factor analysis term is giving you that the other two terms don't give you?
Yeah, so imagine, so we do actually, I brushed this under the rug, but we do include sex as a fixed effect as well, right? But we didn't include age. And I don't know with the iPSC-derived cardiomyocytes, it's not something we've looked at too much, how much age matters. In principle, right, you've wiped away epigenetic memory, and so it doesn't matter. But if age was having an effect, then what you'd hope would happen is one of these X's would end up being some affine transformation of age, and then the W's which are integrated out would be the effect of age on each individual gene expression. So there's no way you could get that part of the covariance structure from it being the same individual or from individuals being related. Individuals being related doesn't mean they have similar age. Does that help? Yeah.
And also, you said technical.
Yes, really we think about it about sequencing depth, library complexity, those kind of things are probably more honestly what it's picked out. I don't know if you know, when Ollie Stiegel and Leo Parts were working on developing PEER, the idea initially was meant to be you do factor analysis, and then those factors are going to correspond to interesting biological processes. So they're going to be, something about immune activation or something. And then what they actually found was most of the factors they were pulling out corresponded to, as you say, technical confounding. And so what people end up doing, right, is associating genetic variation with the residuals instead. So it's the same. So at this stage, we actually want to be able to test SNP gene pairs. And rather than using just this global random effect covariance, we're actually going to use a weighted sum of this covariance with the identity matrix. So what this lets every gene do is choose to what extent it follows this global covariance structure versus doing its own thing and just having additional noise. And you'll see on the next slide why we do that, and it's for computational reasons. So the models we're going to compare, the first one is sort of the baseline model, which is that there's no genetic effect of the SNP on expression. So the expectation of the expression is just given by the fixed effect of concentration. The second nested model is going to be that there's an effect of genotype, but that it's shared across concentration. So the beta here has no dependence on C. And the third is that, and really I should, I guess, have a beta be indexed by G as well here, but in reality, at this stage, we're testing genes independently, so I don't sort of need the indexing over G. And then the full model, which includes the interaction, is to say that there's a concentration-dependent effect of the dosage. So beta now has a dependence on, is indexed by C, the concentration. So this is going to be the math for the talk, which is to show you why using this weighted sum of this global covariance matrix and this with the identity matrix is nice because it gives you flexibility at almost no additional computational cost. The reason that works is that, so imagine we've already done eigen decomposition of the global covariance matrix. So remember, this was estimated across all genes, so I only have one of these matrices. Yeah, question? Yeah.
Question about the method.
Yeah.
So by setting aside the genetic factors from the outset and just learning the random effects, are we putting ourselves at risk of explaining in a way some of the genetic factors?
Yes, definitely. I mean, this in principle, I guess, helps a little bit because you still get to, the A and B here are refit with the genetic terms included. So that potentially helps a little bit. Also, I could make an argument that the genetic relatedness, the kinship matrix is in there, right? But in reality, it turns out the kinship matrix gets almost no weight when you learn the model. So yes, but at least that should make you a little on the conservative side, right? Because it means the random effect variances you learn will be a little larger than they should have been because this variance that could have been explained by genotype now has to go into one of the random effect terms. And those variances being larger will mean you're conservative when you do hypothesis testing. So at least it pushes you in the right direction, I think. Yeah.
Did you actually ever go back and kind of interpret what the factors were? I mean, you're saying you learned they were all technical for sure? Or
No, I didn't look at them. It's a good question.
Okay.
Yeah, yeah, it'd be interesting to check. Yeah, I mean, it's sort of, in a way, nice that you can, I mean, from the point of view of I think it'd be good to look at from the point of view of when you care about the genetic effects, it's sort of nice that you can do this and not worry about it too much. Yeah, so some basic linear algebra from the, once you have this eigen decomposition of this matrix, if you go back to your definitions of eigenvectors and eigenvalues, so Q here is a matrix of eigenvectors. This is a diagonal matrix where the diagonal corresponds to the eigenvalues. Then from your definitions, if you remember these of eigenvectors and eigenvalues, you actually get that you can basically for free get the eigen decomposition of this linear combination with the identity matrix, which you can't do in general when you add two matrices together. And all this does, the Q matrices, the eigenvectors stay exactly the same. The only thing that changes are the eigenvalues are updated exactly according to this linear reweighting. So that's make things computationally very convenient, particularly because when we get to doing likelihood calculations, we will need two things. So we'll need the determinant of this covariance matrix. And if you remember, determinants are just given by the product of the eigenvalues. And we'll also need the matrix inverse. And you can get that directly from the eigen decomposition and in linear time, because all you have to do is get the reciprocal of the eigenvalues. So the likelihood calculation, that multivariate normal, looks like this. Here's that matrix determinant, and here's that matrix inverse. And it actually turns out this simplifies further to this linear sum over these R's, which these you can think of as residuals, except they're rotated or whitened by these Q matrices. So if you look at this expression, this is just Q transpose times Y minus X beta, where here X is the design matrix. And so you can actually make this more efficient by what you actually give the model is Q transpose Y and Q transposed X pre-computed. Since we already have computed, we can compute Q and lambda offline. So the reason this is really helpful is we do this eigendecomposition once, which is cubic in the number of samples, and then we get to reuse these Qs and lambdas to do these linear time calculations across all of we end up doing about 20 million tests because we're testing all SNP gene pairs within a megabase of each gene. To give you a little more on implementation details, so when we actually end up optimizing this function, we have the A and B, so the weights of the linear combination of the global random effect covariance matrix and the identity matrix, and the fixed effects. And we're going to do that using the LBFGS algorithm in Stan. I've been really impressed with how useful Stan is for these types of analyses. If you haven't looked at it, it's a probabilistic programming language. So the idea is you write down your usually Bayesian probabilistic model. It does have the constraint that all of your parameters or variables need to be continuous, although if you can analytically sum over discrete variables, and you can also include them. What's great is you write down your model in this Stan language, which is very easy to read, and then the Stan backend will take that and go and write C++ code for you that calculates both likelihoods and gradients of the likelihood. And then the Stan's team's focus has been on using Markov chain Monte Carlo methods that use the gradients of Hamiltonian Monte Carlo in that setting. But people in David Bly's group have used the same backend to do stochastic variational inference. And you can also, as we're doing here, just use it for optimization. So I think it's very nice that you get to represent your models in this very high-level language, but then you get C++ level computational performance. The only downside is they don't have GPU-based computation yet. It's the sort of thing that it comes up on their forums every couple of years, and someone says, yes, we should definitely do this, and then it doesn't seem to have happened yet. For hypothesis testing, so between those three nested models that I showed you, we just use likelihood ratio tests, and we find that those are pretty close to being chi-square distributed, which is what you'd expect asymptotically. But they're not quite, so I'll talk about how we correct for that. And then the likelihood ratios let us get to p-values, and once we have p-values, we can do we take the conservative approach that's used extensively in EQTL studies, which is to do Bonferroni correction across the SNPs for a gene, and then put those p-values through Benjamini-Hochberg to control FDR. So I think it's something that's people are increasingly aware of, but at some point did not seem to be very aware of. What we want to test for is an interaction effect in a linear model, because we're trying to look for these interaction effects between genotype and doxorubicin concentration. There's no exact permutation test for an interaction term, which really sucks, because usually permutations, the deal is you throw a lot of computation at a problem, but at least you get an exact test. But there's just not a way of doing that in this setting. This nice paper from 2011 that points that out, their suggestion is to use a parametric bootstrap instead. The idea of a parametric bootstrap is that you fit your null model, you then generate new data under that null model, refit your null model and your alternative model, and then ask, do the p-values for that, where you generated from the null, look calibrated? And so using that approach and our procedure, this is showing the number of response EQTLs we find at different nominal FDRs down here versus the empirical FDR that we calculate from the parametric bootstrap. We end up using this setting where the nominal FDR is 5% and the empirical FDR we're estimating at 8.5%. Yes. Yeah, so the asymptotics of the chi-squared, so that needs two things. Well, for it to not be an asymptotic result, you have to have exactly Gaussian residuals, which we quantile or normalize, so we are presumably quite close to that. You also are losing some degrees of freedom to the estimating the random effect variances. And that's not accounted for in the chi-squared. The degrees of freedom we use are just from the design matrix. And it's an open question. It's an open question. I think there's also not a good answer of trying to calculate what's the effective degrees of freedom in a mixed model. And so that's effectively, I think, where the difference comes from. But it's also probably counteracted somewhat by the fact that our multiple hypothesis correction is a little conservative, right? Doing Bonferroni and then Benjamini-Hochberg. So there's some debate about whether iPSC-derived cardiomyocytes represent anything meaningful in vivo. One way we wanted to look at this computationally was to ask whether, so comparing model 0 to model 1, you can think of those as sort of marginal effect EQTLs rather than interaction effect. And then we can ask if those are enriched across different, the EQTLs from different GTEx tissues. So this is using EQTLs called by the GTEx consortium. And when initially I did this, I was like, oh no, this doesn't look too great. So I've highlighted here the two heart tissues, and the metric I'm showing on the x-axis here, stories pi1. This is an estimate for EQTLs significant in our data of how many of those look like they replicate across these different tissues. And while the two heart tissues are pretty close to the top, there's some weird things like thyroid and tibial artery that are higher up. It turns out one of the challenges with using these GTEx summary statistics is that there's quite significant variation in sample size across the tissues, so there's just different power, which completely confounds this analysis. We have a different approach where we have a method which I can go into afterwards if you're interested, but it basically tries to estimate mutual information between whether a SNP gene pair is an EQTL in our data versus an EQTL in the GTEx data. And when you do that, that implicitly accounts for the differential power between the GTEx tissues, and then you see the heart tissues popping out on top. So while the GWAS that I told you about at the beginning of the talk were not very convincing, our hope was that there's still some meaningful signal in there if you look at further down the p-value list. And so the enrichment I'm going to show you here is looking at significant EQTLs, both in GTEx and in our data, and then asking for those what proportion, looking at the number of the GWAS p-values corresponding to those SNPs that are under 0.05. So, pathetically non-stringent threshold on the GWAS side of things, but that's just because the GWAS data is so underpowered. So this is looking at GTEx EQTLs from just 3 tissues, and you see no enrichment. If you look at our induced, our iPSC-derived cardiomyocytes at baseline, you don't see any enrichment. If you look for main effects, which is what I showed you the enrichment for on the previous slide, you see no enrichment. If you start looking at EQTLs that, combined here means comparing model 0 to model 2, which is saying, is there any effect, main or interaction, for this SNP? Then you start to see some enrichment. Either here is, instead of doing that statistically, just compare model 0 to model 1, which gives you main effect EQTLs, compare model 1 to model 2, which gives you interaction effect EQTLs, and just take the union of those. Then you see a slightly stronger enrichment, but it's comparable. And then if you look at the response EQTLs, so the interaction effects, then you see the strongest enrichment. And the last column here, I can't show you until I talk to you about using allele-specific expression. So we and others have shown that it's possible to use allele-specific expression to improve power to detect both regular EQTLs and gene by environment, so interaction EQTLs. And the rationale for this is if you have a heterozygote regulatory SNP in an individual, and you need to also have a heterozygote exonic SNP, then in the setting where there's an environmentally or stimulus-dependent transcription factor that binds at that regulatory SNP, then you'll see allele-specific expression as a result of the differential binding between the two alleles in the single individual. The example I'm showing here is this P53 that I mentioned from the functional enrichment analysis. Mathematically, the way we handle this, so Y now, rather than being the quantitative, the continuous measure of gene expression, this is now minor allele count for an exonic SNP. N is going to index over individuals again. K here is indexing over the SNPs exonic SNPs in a gene, and C is concentration. BB here is the beta binomial distribution. So you could use a binomial distribution, you would think, to model the proportion of RNA-seq reads that you're seeing coming from the minor versus alternative allele. It turns out you really need to account for overdispersion as well, and the beta binomial lets you do that through this concentration parameter gamma. The parameterization I'm showing here, R here is just the coverage, so the total number of reads mapping to that exonic SNP. And then this term is sort of like a logistic regression, so giving the mean of the beta binomial. So sigma here is the logistic function. Mu here is what we use to account for the fact that whatever you do at the mapping stage, you tend to still have some reference mapping bias left over. And the most important term here is this phi, which is the phased heterozygosity. So what this is it's a variable which can be -1, 0, or 1. It's 0 if the regulatory SNP that we're testing is homozygous. We still include those samples because it still helps us fit this concentration parameter gamma. It's +1 if the heterozygote regulatory SNP, if the alternative allele is on the same chromosome as the alternative allele for the exonic SNP that we're looking at, and so they're in phase, and -1 if they're in the opposite phase, so on the alternative alleles are on the opposite chromosomes. And again, we're going to have to specify these three nested models corresponding to there being no genetic effect, which means this beta term is 0. There being a main effect of genotype, which is just that the beta is non-zero, but doesn't have a dependence on concentration, and this interaction model, which allows unconstrained beta C. As you might guess, I fit this using STAN and LBFGS. So in an ideal world, you would have one model that would look at both the total expression and allele-specific expression at the same time. When I've tried to do that, it's really hard to get comparable effect sizes between the two models, the two data types, particularly because we're doing this quantile normalization of the total gene expression, which seems to be really helpful. So what we actually end up doing instead is just adding the log likelihood ratios from the total expression model and the allele-specific model, And you can do that and still know what the null distribution is because you're just adding variables which are chi-squared distributed, and that just gives you another chi-squared you just add the degrees of freedom from the two tests. When we do this, we get the strongest enrichment. We actually only find on the order of 30% more response eQTLs, but we do also, along with that, see an improvement in the enrichment that we see. We've also looked at alternative splicing in this data set and the changes with doxorubicin. Just to remind you, alternative splicing is this process by which there can be different choices in the isoforms that are produced by the splicing machinery. Here I'm just showing an example where exon 2 of this gene is included in isoform 1, but it's not in isoform 2. And we can quantify that using that variation using RNA sequencing. And we're going to talk a little bit about how we use Leafcutter to do that. So the previous work we've done using for splicing quantification, the main idea was to, rather than traditional approaches which have tried to quantify exonic percent spliced in, so how much is an exon included in transcripts relative to its neighbors, or to quantify at the isoform level, so how much is each possible transcript for a gene expressed. We quantify intron excision rates, so how much is each intron used. The nice thing about that is intron usage rates. Introns themselves are uniquely defined by a spliced read. So if you give me a spliced read from RNA-seq, I can tell you exactly what region of the precursor messenger RNA was spliced out. That gives you very easy quantification. Just count how many junction spanning reads there are. And it makes you less reliant on existing gene models, which you're very reliant on for the exon or isoform-based approach. And you can model complex splicing events. What this looks like for a little example I'm showing here, where you have some gene. Black here are constitutive exons, and white are alternative exons or a 5' extension here. The splice reads you might see in this region might look something like this. And what Leafcutter does is to construct this graph where the nodes of the graph have starts and ends of introns or junctions. And we put an edge in the graph wherever a spliced read, so a junction is observed between a donor and receptor. And then we define these splicing events as connected components in this graph. So what this would end up being in this setting would be that you have one component to the left of this middle constitutive exon and one to the right. Then the quantification we use is just the number of junction spanning reads supporting each of these junctions relative to the other junctions in the same splicing event or component. We do differential splicing analysis using a Dirichlet multinomial generalized linear model, which lets us model over dispersion and this idea that these junctions are being chosen relative to each other in a single component. In this data set, we see with the large sample size we have, a large number of the splicing components that we detect being differentially spliced with respect to doxorubicin. And I'm showing one example here of this gene involved in apoptosis. Sorry, this figure's a little bit small, but it's also in the paper if you want to look at it. What you see here is increased usage of an alternative exon with the moderate doxorubicin concentrations. And interestingly, you only see that in the presence of a minor allele for a specific SNP. So this ends up being a response splicing QTL rather than a response expression QTL. The overall pattern we've actually noticed in this data with regards to RNA splicing is that doxorubicin just seems to reduce overall splicing fidelity. So maybe the most clear way to see that is we clustered introns into those that are clustered introns by their response, their relative intron excision rate across doxorubicin concentrations, here into 8 different clusters. And what you see is, and it's clearest for these first 2 clusters, this first cluster is introns that are being used less frequently with increasing concentration. The second cluster is introns that are being used more with increasing doxorubicin concentration. And then if you look at how often those introns have ends that are annotated in gen code, what you see is these introns that are being used less with increasing concentration are introns where both ends are annotated, so they're sort of canonical normal splice events. And then the second cluster where you're seeing increased usage are introns where predominantly only one end is annotated, so at least one cryptic splice site is being used. So, and we've looked at this in a few other ways as well. You can do an analogous analysis for these splicing QTLs and the enrichment in GWAS. And again, we see a similar pattern that the response splicing QTLs are where the strongest enrichment is. Something else we've been able to look at in these iPSCs is derived cardiomyocytes is the level of cellular damage measured by cardiac troponin, which is is found in media after the cells lyse. This is showing the cardiac troponin level at different concentrations, and each line here corresponds to a different individual. So where you're seeing these higher concentrations of cardiac troponin corresponds to individuals where more cellular damage is seen in response to doxorubicin. We initially tried to associate into the variation in cellular damage with the transcriptomic response by defining a overall transcriptomic response by starting with that principal components analysis I showed you at the beginning, and fitting what's called a principal curve through that data, through that trajectory. And then for every sample, you can define a position along this trajectory and ask for any given individual how fast is that individual traversing this trajectory. When we do that, so that transcriptomic response measure for each individual is on the x-axis here, and the rate of the cellular damage, so this is just the slope of this data for each individual. You see that there's actually a negative correlation between the cellular damage and the transcriptomic response rate. And I should say this is the initial transcriptomic response rate, so this is how fast are you initially traversing this trajectory. So it's really interesting that this is a negative correlation, because what this suggests is that the individuals who are responding most strongly in terms of the transcriptomics are actually the ones where you see the lower cellular damage, which is suggestive that actually the transcriptomic response that you see in the cells, at least initially, is protective against cellular damage. Something we've done using the cardiac troponin measurements is to do what's effectively a transcriptome-wide association study. So for those of you who aren't aware, the idea with these is to leverage the situation where you have a GWAS cohort where you have lots of individuals but no molecular measurements, but you do have some trait of interest, and a reference cohort where you have molecular measurements like expression, and to use learned predictive models of expression from cis variation in your reference cohort to impute molecular phenotypes into the GWAS cohort. What we're actually doing here is using the same cohort on both sides. But what this lets us do is worry less about reverse causation. So if we use predicted gene expression values and associate those with the level of cellular damage, then we can worry a bit less. That's effectively getting the genetic component of the gene expression. And then we can worry a little less about reverse causality, so that the gene expression measurements that we're correlating with cellular damage are less likely to be downstream effects of cellular damage. So we build lasso models of gene expression from concentration and genotype. And for the around 3,000 genes where we see a significant genetic component, we then associate those with the level of cellular damage. And the optimal cross-validated lasso model ends up with around 100 genes in it. I think this is cool because if you try to do, this is effectively getting at something that looks a lot like a GWAS for cellular damage, but we have 45 individuals, right? So there's no way we could actually have done a GWAS. But because we have this intermediate level, this intermediate component of gene expression, we're actually able to find a meaningful model from genotype to cellular damage. We've also, to sort of think a little bit more about in vivo effects, we've used some published data where they looked at iPSC-derived cardiomyocytes and doxorubicin response in patients who had had chemotherapy with doxorubicin and who had or had not resulted in, which had or had not resulted in cardiac side effects. And using that data, we're able to build a predictive model from the expression response to ACT, anthracycline cardiotoxicity risk. And we see that our measure of cellular damage is correlated with that predicted ACT risk. If you're interested in reading more, the paper came out in eLife last month, and there's lots of supplementary figures for you to look at. And code for the method is up on GitHub, and we've tried to be good about putting the analysis up as well. I was going to talk about future work a little bit, but I'm also out of time, so I can wrap up also.
Yeah. I think we should wrap up, and then if you want to show a little more in the discussion, future work, we can do it then.
Yeah, so I'll leave that there. Thank you.